Hello everyone, it is Sabah Al Hassan from Beirut Arab University, Faculty of Health Science, Medical Laboratory major, and today my topic will be about smoking. So first of all, it's well known that smoking is one of the major leading cause of death throughout the world as it causes sickness and disabilities and kills approximately 5.6 million people. Moreover, 1 in 3 cancer deaths are caused by smoking, since one single cigarette contains more than 4,000 chemicals, 60 of which are known to cause cancer. Smoking kills people according to their age and gender, and as we can see, that males has higher proportion of death compared to females. Most commonly, smoking leads to lungs, heart, and digestive system damage, in addition to the ingestion of harmful chemicals present in cigarettes, as well as spending a lot of money on cigarette smoking. Smoking harms every single organ in the body, reduces your health, and may cause diseases. But as a medical laboratory student, I will be talking about the effect of smoking on laboratory results. Both active and passive smoking have a significant effect on hematological values such as hemoglobin levels, red blood cell and white blood cell count, hematocrit, MHC, MCHC, and mean cell volume. Smoking 10 cigarettes a day results in slightly higher hemoglobin, HCT, and MCV. This is probably a consequence of the elevation of levels of carboxyhemoglobin in addition to its accumulation in blood, as after a single cigarette, the level of hemoglobin increases by about 1%. This is associated with polycythemia, which respectively leads to high hemoglobin levels and hematocrit. On the other hand, total leukocyte count increases largely as a result of an increase in the neutrophils count. Also, smoking may cause an increase in CD4 lymphocytes and total lymphocyte count. To add, smokers tend to have higher platelet counts than non-smokers, but these counts decrease rapidly on cessation of smoking. Other blood counts are affected by smoking, either by an increase in parameters or decrease in parameters. Plasma viscosity, haptoglobin, fibrinogen, beta thromboglobin concentrations, as well as VWF level, ESR parameters increases. However, plasma volume and protein S parameters decreases. Smoking leads to hypoxic conditions in the blood. This adversely will affect your body. Now, to talk about the most common test done in order to detect smoking, which is the nicotine test, and we should mention that nicotine is a highly addictive chemical. This test measures the level of nicotine or chemicals that smoking produces in your body. In addition, it can detect if you smoke or use other forms of tobacco. And we know that all forms of tobacco have nicotine and cotinine, which is a chemical produced by the breakage of nicotine in the body. Now, regarding the sample required, we should note that sample taken depend on the type of test done. So it can be either blood sample, random urine sample, saliva sample, or even hair sample. And to talk specifically about each test, first we have the blood test, where a lab technician will insert a needle into your vein to collect the sample. This test is a very effective approach and can detect the tiniest traces of cotinine in the bloodstream. However, the urine test, it is said to be the standard and most common used for testing of nicotine. It can be done by anyone at home. All you need to do is buy over-the-counter strip, dip it in the urine for at least 5 minutes, and read the results, or you can submit the sample to the nearest laboratory. On the other hand, we have the saliva test, which is similar to the urine test. Saliva is required to be soaked in a strip. It is considered as the most accurate and most preferred method to test for nicotine. And finally, we end with the hair test, which is a reliable way to figure out long-term use of tobacco products and can be very accurate for 1 to 3 months after you stop smoking. How does the test work? There are couples of ways to test for nicotine and cotinine, either by qualitative testing or quantitative testing. Qualitative testing simply looks for whether or not you have nicotine in your body. However, quantitative testing measures the concentration of nicotine or cotinine in your body and gives information about your tobacco habits. And finally, the test results. What does the test result means? The test results can be reported as positive or negative or as a given level. A high level of nicotine or cotinine indicates active tobacco or nicotine product use. However, moderate concentration indicates that the person has not had tobacco or nicotine for 2 to 3 weeks. A lower level may be found in a non-tobacco user who has been exposed to environmental smoke. And finally, very low to non-detectable concentrations may be found in people who have not used tobacco or nicotine and have not been exposed to environmental smoke, or in tobacco or nicotine users who have refrained from tobacco and nicotine for several weeks. And at the end, you should stop smoking for better health. Thank you for watching.